to Dr. Daniel Schramek. Did I get that? Okay, thank you. Who is a principal investigator at the Lunenfeld Tannenbaum Research Institute and an assistant professor at the Department of Molecular Genetics at the Faculty of Medicine of the University of Toronto. He trained in Europe, Australia, and the US, receiving a BA and a Master's in Science and Molecular Biology, a PhD in Genetics, and an Executive Master of Science in Technology and Management. He trained as a postdoctoral fellow and Emerald Foundation Young Investigator at the Rockefeller University of New York. And Dr. Schramek's research focuses on leveraging functional genomics to make major advances in treating human cancers in a personalized and highly specific manner by characterizing why a tumor develops and identifying specific vulnerabilities of brain, breast, and pancreas cancers. I also want to mention that Dr. Schramek is one of our 2021 Brain Tumor Foundation of Canada research grant recipients. And the research he looked at and what he's going to be expanding on today is low-grade gliomas, are brain tumors that are at first very relatively low-grade brain tumors and not harmful and tend to grow unrecognized over years and sometimes decades to a size that causes headaches and seizures. Unfortunately, almost all low-grade gliomas eventually progress to high-grade glioblastoma, and Dr. Schramek will share his research on this topic. And the title of his presentation today is Inherited Factors that Predispose to Brain Cancer. Welcome, Dr. Schramek. Thank you so much for joining us today. And the stage is yours, so to speak. I'll stop sharing my screen so you can share yours, and we'll go from there.
All right.
I'm going to mute myself and take my video off, but if you need me, I'm just a click away.
Terrific.
Perfect.
First, before I start sharing, let me just say how grateful I am for everybody attending, how grateful I am for the foundation. It makes a real change to our lives, having access to a foundation, to patients, to patient advocacy groups, to and also for the money and the grant which the foundation is actually raising. Since many years and so efficiently. Now, my story is also quite unique and has to do with brain cancer, because I'm actually not necessarily, or I wasn't a brain cancer researcher. My background was more in breast cancer. I had a neck cancer. Are those cancers which form in the oral cavity and the throat. And then we did a little bit of pancreas cancer and lung cancer. But I, till 2015, have never worked in the brain cancer realm, which is really quite different to these other epithelial cancers. The reason I started with brain cancer was because my twin brother was actually diagnosed with a low-grade glioma at the very week when I accepted my job here in Toronto. And my brother, and as you can maybe hear from my accent, is Austrian. He lives in Vienna, and I'm obviously very far away. So the only thing I could really do is just to read up and get myself interested. And doing that, I realized that there are many things which are not really addressed right now in this brain cancer, and thought that we might actually have a way to help the research, at least, given that I couldn't be with my twin brother, at least for extended time period. So that is obviously a quite private journey, but it gives me insights into the disease, which usually researchers not necessarily have. And I think that sparked interest, a certain amount of perseverance to overcome hurdles, and an understanding of what it actually means to be a caregiver or a relative to somebody suffering from this disease and thriving. Lastly, before I start my presentation, luckily my brother is doing really fine. He had quite a journey with open surgery, with while he was still awake surgery, then radiation, then chemo, but right now he's doing actually really quite fine. Having said that, what I wanted to do today, and I spent really quite some time yesterday to put together a presentation, is give you a little bit of an insight of how we address brain cancer in the lab. Now, that's obviously really quite specialized, and we use very difficult and funny words, and have got different paradigms and experimental systems which are really quite hard to understand. I give you one example. If I talk to my wife, who is with me since 15 years, she's getting lost really quickly, despite the fact that she hears about science every single day. So what I tried to do is to really break it down into what I hope is the easiest to understand schematic talk. And the only thing which is important for me today is that you just get an idea of what we do in the lab to really dig into this disease, understand the disease, and probably come up with ways to treat this disease. Now, with that, I will share my screen. Maybe one more thing. I don't know if this is actually possible, but if things get too complicated, and I'm trying very hard that they are not complicated, maybe just raise your hand and interrupt me. And Janice, I don't know if this is possible, but let's just get started and then we see where we are.
Yeah, people can add comments in the chat, Daniel, if they have any questions, or we can clarify some things during the Q&A for you as well, for them as well.
I like that. All right. So I think everybody here is very much, can you see my presentation?
Yes.
Oh, very good. Everybody here knows that brain cancer is a huge problem. It affects 3,200 Canadians every year. And without going too deep, it is obviously also a very deadly disease. And we have really, as a research community and medical community in this disease, not made too much progress over the last years. And that is multifactorial. One of the main reasons is that brain cancers are usually diagnosed relatively late. And that's certainly one of the problems. However, I really do think that there is hope because there's more and more research. And I'll give you one snippet to convince you of that. In breast cancer, the survival 20, 30 years ago was really, really low. We're talking about 20% at 5 years. And breast cancer was basically the poster child of cancer research for the last 30, 40 years. And now survival is up to 90, 95% at 5 years. So a huge change, and all that is basically because of research and medical teams working really hard on that problem. And I actually hope that the same thing is going to happen for brain cancer. Now, sorry, too fast. The agenda for today is I thought I might talk you through two different stories. The one is revealing the genes that cause glioma, and that can be genes which are picked up as a mutation within the brain tumor, or variants which are really quite benign, but can in certain settings predispose to brain cancer, and these are inherited. And the second story is this inherited predisposition to glioma development and how we can actually get a handle on who is predisposed and what does it actually mean, and go from there. Now, brain cancers arise in the brain. That's something you all know, but very, very likely they actually arise in stem cells. And in the brain, the stem cells are actually lining the ventricles. So the ventricles are these cavities in the brain, which contain the cerebral fluid, where the brain is basically bathing in. And these stem cells are, something is wiggly, are lining these ventricles all around, okay? So, and I, as a schematic, just drew them here, right? You can imagine that this is the cavity where the spinal fluid actually swims, and these are the stem cell enriched niches. Now, I'm having a problem with my, this works now. Now, brain cancer arises when cells accumulate mutations that inactivate genes that usually tell a cell to stop growing. Okay, these are very important fail-safe mechanisms for a cell to stop growing. And we call them tumor suppressor genes. They suppress the tumorigenic potential of a cell. Now, when tumor suppressor genes are mutated, then cells start to divide uncontrollably. And eventually they become cancerous. Now, in brain cancer, compared, for example, to white skin cancer, we don't see these lesions. In the skin, which is exposed to the outside, people actually pick up these pre-malignant lesions really, really quickly, but they're really concealed in the brain, and that's one of the problems. But then they usually start to grow and form a tumor. Now, sequencing studies have identified mutations in the DNA of brain tumors. Okay, there seems to be a problem. And there are usually hundreds of these mutations. It's not one mutation, it's really hundreds of these mutations. sorry, this has actually got a problem here. Doesn't matter. So this is actually to show a DNA strand. It doesn't matter. These mutations actually happen in the DNA. And usually we've got hundreds of mutations in genes, which codes for protein, and thousands of mutations, which are actually in the non-coding regions. So there are thousands of mutations. And the real question is, which of these genes acts as tumor suppressor in the brain, and which of those are actually responsible for generating tumors? Now, my lab, we are trying to figure out what these genes are, because these genes and these pathways might be important to target for therapy. Now, obviously, you cannot do these experiments in humans because it would be unethical to ask which gene, when mutated, causes breast cancer. So we turned to mice, and mice are very, very good model organisms to study different cancer diseases. And we established very complex, innovative mouse models so that we can test many, many genes within one single mouse. So what we do is we take CRISPR-Cas9. This is a new gene editing technology. You can think of it as molecular scissors. And then we disrupt the DNA by cutting the DNA in the genes which we find mutated in human patients. And that basically allows us to model the mutations which we see in humans in mice. And then we can study their role in mice. Now, we don't do this with just one mutations. We do this 100 to 1,000 times in different cells within the brain. And that allows us to actually analyze many, many, many genes within one single mouse. And that's a very new technology. Now, eventually, a few of those cells, when we hit the right tumor suppressor, will go on to form a tumor. We can then excise this tumor, deep sequence the tumor, and figure out which mutation actually caused the tumor. Now, this is now very, very interesting because the same mutations which causes tumors in humans cause tumors in mice. So we have got now a model system which we can use to A, figure out the genes which cause tumors, and B, we have got a model system where we can ask, what do these genes do? And what do I, as an experimentalist, have to do to stop these tumors with these specific genes? So using this, we basically set up the first phaseful low-grade glioma model. And now we can go on and test different drugs and different genes and ask which of these drugs or genes can actually stop the growth of these tumors, and then hope that these tumors actually even shrink or disappear. Once we have found these genes or these drugs, we can actually ask and see, does this hold true in a human setting? Now, we would not move on to a patient right away. We would move on to cellular systems of those tumors, or stick these human patient tumors into mice and repeat all this. But conceptually, that's the way to go. Now, and then the hope is that these drugs might also work for human patients. Now, in a second story, I wanted to tell you a little bit about our work, where we're not only focusing on the genes which are responsible for delineating or causing tumors within the tumors itself, but also about factors which we might inherit from our parents and that predispose us to develop tumors. Now, let's think a little bit about inheritance and cancer. All cancers have some components which are heritable. In breast cancer, we know that 5 to 8% are actually completely inherited. And these are patients which carry, for example, germline mutations in genes such as BRCA1. And they will very likely go on to develop breast or ovarian cancer. We know these high-risk patients, and we have learned how to actually deal with these patients and help these patients in their clinical decision-making over time. But much of the inheritance of other cancers is still relatively unknown. And it's important to figure that out, because that might actually help us to diagnose patients, for example, much, much earlier, and have them under much, much more surveillance. And surveillance is something which is done quite well for breast cancer and prostate cancer, but not really for brain cancer. So before I can tell you a little bit about inheritance, we need to think about the genetic code. And if you think about the alphabet with the 26 letters, the genetic code is basically also an alphabet, but only has 4 letters, A, T, Z, and G. And these 4 letters make up words, but only that we call them genes, okay? And then humans are made out of a book, so to speak, the story of development and body structure, which is made up of 3.2 billion letters and about 20,000 words, 20,000 genes, okay? Now, while copying this book of life, you basically have slight variations, small variants happen, and usually they're completely benign. You can think about it as an example of the word tumor and tumor, spelled once in British English and once in North American English. That means the same, but there's clearly a slight variation, right? One has a U, the other one doesn't. But it doesn't change anything to the meaning of the word. It doesn't change anything to the meaning of the book. And that's most of the time, there is no change if you have got a slight variation. However, sometimes this actually causes a change, and it's extremely hard to figure out whether or not a misspelling causes a change or not. Now, the other thing which we need to consider for this story is what is the difference actually between the normal tissue and a tumor? A tumor is still your own tissue, right? It's very different than a bacterial infection where you take an antibiotic. There you have got a microorganism invading the body and it's foreign. But a tumor is self. It's part of your own cells, right? But there are differences, and it's important to figure out what these differences are. So I already told you about the concept of these tumor suppressor genes. These are genes which evolved throughout time to actually make sure that the cell does not transform, right? You can think about them such as brakes within a car. And then you have got things which we call proto-oncogenes, which can drive tumorigenesis. You can think of those like the gas pedal within a car. These things are usually very, very important for cellular growth during development when you grow up. But at one point, you need to be in check and you need to step off the gas, because if not, you have got uncontrolled proliferation. Now, in cancers, usually what happens is that you need to do 2 things, and that's something we call the 2-hit hypothesis. You need to break the brake, you need to get rid of the brake, and you need to step on the accelerator and turn on an oncogene. And the way to think about this a little bit better is if you think about a car and you take the analogy of a brake and an accelerator, you can think of a normal car going its normal speed, but it doesn't hit the wall of cancer. If you now have the first mutation, and for example, you have a mutation in a jammed accelerator, the car will speed up, but it's still, you have a handbrake, and the handbrake will basically stop the car. However, if a second mutation is picked up, and you also have a mutation now in the brakes, then this car will accelerate and really hit the wall of cancer. So several things have to happen. You have to get rid of your tumor suppressor, and you need to activate the oncogene. You need to accelerate, and you need to break the brakes. Now, how do we find this second hit, and how do we find factors that can predispose and accelerate tumorigenesis? And these can be either the brakes or the accelerator. Now, how can we determine the second hit? Again, we cannot do these experiments in humans. That's unethical. So we turn again to mice. And what we did was actually surveying hundreds of low-grade glioma patients and asking what is different not only in their tumor, but what is actually different in their germline compared to healthy individuals which don't have low-grade glioma. And that was done over thousands and thousands and thousands of patients. And what we found is that one single nucleotide, one single letter out of the 3.2 billion letters is changed from an A to a G Now, this letter is usually found in a normal population. It's a completely normal variant. It's just not very common. It happens in about 0.01% of people on Earth. However, in low-grade glioma patients, 40% out of a sudden show the G allele. They show this altered nucleotide, which is so rare within the general population. And if you see this increase, this is a 600-fold increase in the likelihood of patients having this one letter. That basically indicates that at least this letter or something, some gene surrounding this letter might predispose to brain cancer. So what we did was actually, again, using these CRISPR-Cas9 gene editing gene scissors and changed exactly this one letter in these 3 billion letters of a book, and then asked, does the mouse, which is spelled tumor or tumor, have a different prevalence in generating low-grade gliomas? The way we do this, just to give you an idea, is actually, because we need to get into these mice very early, is to take a pregnant mama mouse, anesthetize the mama mouse, make a midline incision, then we exterior the embryos, and under ultrasound guidance with a micromanipulator, with a needle we have to blow ourselves and actually sharpen with a diamond sharpener, inject the embryo. Here, this is actually the picture of our mouse ICU unit where we do these surgeries. These are live survival surgeries. Basically the same thing which we do with patients, right? Here's the tail of the mouse. Here's the little embryo sticking out of the belly. And here's the ultrasound. And then here you see the ultrasound pictures blown up. And what we try to do is actually take a needle and go into the uterus and expel a little bit of virus, 62 nanoliters, nothing, you cannot even see it, into the ventricle of these mice so that we infect these mice. And to show that this works, we basically put green fluorescent protein into these mice, and you can appreciate that the brain of the mouse out of a sudden lights up green, okay? So this technology works, and with this technology, we can now actually take these CRISPR gene scissors, put them into the brains of mice, and very distinctly change one nucleotide. And when we do this, and here is just an example where we targeted two very common brain cancer genes, NF1 and P53, in the control mice, they don't develop any tumors. This is a completely normal brain. But if we now actually use our gene scissors to cut those genes and mutate these genes, the same way we see it in patients, out of a sudden you can appreciate that you have got this huge tumor in these mice. And using this technology, we could actually now show that this one letter change, which we find in patients, but hardly ever in the general population, is actually really predisposing to the generation of low-grade gliomas. And that is really quite meaningful because that happens in 40% of patients. And we also have got a way of either now using this information to either diagnose patients much earlier, or if you see, for example, a flare in a MRI of a patient and you're not sure, this knowledge might actually help you to decide whether or not to start treating these patients much earlier than you would without. And then the chance of actually curing these patients is much higher. So that's where we're going right now. This still needs to be tested. Importantly, these findings were just published in Science, which is one of the two most prestigious journals. And we are very happy that that worked out. And this was actually with the help of the foundation. Now, what's next? My last slide. I think now we need to use our systems to not only ask what causes cancers, which genes, when mutated, cause cancer. I think now we need to use our technologies to actually ask within our live models, which genes, when destroyed, stop those cancers to grow. And that's the big next task for the next 3 to 5 years. And with this, I really want to acknowledge the people doing this work. You can see that there are plus 60 people working on such a project. Foremost, I want to thank Connor Yankers, who is the first author of this paper. He worked very, very closely with the Mayo Clinic, Kirsten Drucker, who works in the lab of Robert Jenkins, who is a MD and professor for epidemiology and cancer genetics, specifically in brain cancer. But it was also a terrific collaboration with our colleagues from SickKids, Peter Turks, Michael Taylor, Akram Gelarizadeh, and other clinician scientists with Michael and Peter and Michael Wilson. And then a lab with in Vienna, Ulrich Elling, who helped us a lot with the technology side of all this. Now, lastly, and I cannot emphasize this enough, thank you so much to the hundreds and thousands of patients who were willing to actually donate their tissue after surgery for these studies. It is imperial to get these things. Without the patients and without our, what I call patients collaborators, we would be stuck in a black box and not be able to move. So these are extremely, extremely important. I really want to thank the donors and the fundraisers. I cannot emphasize enough that philanthropic money enables us to start working on ideas which are really high risk, such as this model of destroying genes within the brain of mice using gene scissors, which basically came out of yogurt research. It needs this spark of unrestricted funding before more conservative funding systems start to work, which is then the government of Ontario, the government of Canada, and international funding bodies. But you need something in the beginning, and that's really what donor money enables us to do. And with this, I'm going to finish, and I am very happy to take questions.
Thank you so much, Dr. Schramek. That was so interesting, and you really did explain it in a way that was really easy to understand. One person is asking, the SGNF1 and SGTRP53 genes, are these analogous to the BRCA gene for breast cancer? And are there opportunities for gene therapies with this research along with drug treatments?
So, excellent question. Yes, they're very analogous. We have got germline carriers of TP53 mutations, and we've got germline carriers of NF1 mutations. Both of them are predisposed to getting cancer. So it's a familial cancer syndrome. One is called Lieframeni, the other one is called neurofibromatosis 1. Both of them are also predisposed to brain cancer. And interestingly, once you have a P53 germline mutation and you pick up an NF1 mutation in the brain, tumors happens, and vice versa. So there's a real interplay between those two genes.
Okay, thank you. Somebody had asked, and I had answered their question kind of privately, but we're asking if this, if meningiomas were falling under this research, but I explained that meningiomas don't fall under the low grade low glioma umbrella. But the follow-up question was, would it be possible to use this type of research or this type of technique with a meningioma type brain tumor? Okay, thank you. Somebody asked, what would cause someone to have an MRI before tumor is discovered? I know what I would say, but I'll let you answer that.
So many patients undergo MRIs after an accident just to see if there's concussion. Many patients undergo MRIs because they have got a headache and then don't know what happens. So there are many incidental findings which many times are just ignored because it's not what you would expect. But those patients, to follow them up is actually really quite important.
Yeah, exactly. Somebody said, so if a family member has a history of brain cancer, other family members should be aware, ? So is there a way to be tested before any symptoms present themselves?
So again, that would be for this very, very minor and small and rare sliver of society, which actually carries this genetic variant. And in the near future, we're going to have much more genetic history and ideas and actually know what to do with genetic information. Because right now, we can read genetic information really easily. It cost $3 billion 20 years ago to actually sequence a human genome. Right now, we can do it for 1,000 and soon for $200 within an afternoon on a lab bench. So that's going to be standard. The problem is understanding what it actually means. And that's exactly what my lab is doing, right? You need to really understand what every single base pair means before you can do any interpretation. And so I think we are heading there, but we are not quite there yet.
Right. Okay. Thank you. And lastly, my husband passed away with a grade 4 glioblastoma within weeks. Do you have access to the tumor board? We were not even asked to be a donor.
So these things have to be always set up well in advance,
Right?
right? So this is not something where it takes years of preparation, and then it takes a lot of patient consent. And that cannot be done when a patient progresses so quickly. And I'm super, super sorry to hear that.
Yeah.
But we have got, right now we are not in a dear situation of getting enough samples. Right now, the bottleneck is somewhere else. Having said that, if there is a major center, usually or very, very often, there is consenting and biobanking of materials to save for the future. And that's also very important for us.
Okay, thank you. Sorry, I think there's a couple other questions that have popped up here. Can brain cancer genes be passed on over several generations in a family, father to son, for example?
Yes.
Yeah. Do genetics professionals have access to tests that you are working on?
So right now, how science works is that you first have discovery science, and that's where I am, right? It's very, very early. And then you start developing clinical tests, and from these clinical tests, they can become implemented. And that's a journey which takes a certain amount of time, depending on what the test is and what the implications are. For what I presented today with these germline mutations, that's actually really quite easy and wouldn't take a lot of effort and a lot of time to implement. But we are just in the beginning to actually start doing that. So yes, soon, I hope that this will be implemented.
Perfect. Thank you. I'm just sending a quick message to my colleague here. Okay, perfect. I think that's, one other question that popped up, might be a hard one to answer. My daughter died of a glioblastoma, so sorry to hear that. Her 6-year-old son has headaches. Would this be enough for an MRI request?
So I would say no. A clinical, so I'm a geneticist. I'm not a doctor. I'm a PhD and a professor for medical genetics. So that's really the best question to ask for the MD specialists. From what I can see right now, headache, everybody has headache. And depending on what the symptoms are, how long lasting it is, the medical MDs will really know what to do. In 99.99%, it's nothing, because brain tumors, especially in young kids, are very, very, very rare. But if it's persistent, it might be a good idea for many, many other reasons to do further diagnostics, and the MDs will decide what's best. They know everything really quite well.
Right. Thank you. I'm just going to ask one more question. Are there opportunities for gene therapies with this research along with drug treatments?
Gene therapy in cancer, the way you probably envision it, is very difficult, because what you're thinking is, can we take a tumor and then repair the gene which is destroyed in the tumor itself?
Right.
Problem here is you would need to be extremely efficient and really reach every single cell to 100%, and then repair the defect to 100%. If not, and if there's only one cell left, it doesn't help you, because that cell might grow out to basically cause problems down the line.
Right.
Gene therapy for tumor therapy, I think, is not really gonna work in the way I described it right now. Okay. Gene therapy is used a lot to modulate T cells, so immune cells, and then put them back into a patient to aggressively go against the tumor. That works. But gene therapy for therapy of cancers itself, I don't think so, but who knows what the next 5 years of research bring.
Right. Perfect. Thank you so much, Daniel, for joining us today. There were a couple other comments and questions that have come through, but what I would like to do after this webinar is kind of sift through them to see if they were similar to the questions we asked. If it's okay, I might follow up with an email with a couple more questions, and then we might be able to include that information in our email follow-up with the recording for everybody. Thank you for all the great questions and comments, everyone. And thank you for joining us today, Dr. Schramek. We really, really appreciate your time and energy and expertise around this. And we'll definitely be in touch.
Absolutely. Thank you so much.
You're welcome. Thank you. Okay, Jen, you are going to be up